All right, hello, welcome. My name is Katherine, and I'm a senior biology major here at UCR, and I'll be presenting on my honors thesis, an evaluation of food webs in the Sierra Nevada, under the guidance of my faculty mentor, Dr. Kurt Anderson, who is a part of the Department of Evolution, Ecology, and Organismal Biology. Freshwater covers a tiny area of the planet's surface, 0.5% to be exact. But it's vital for our economies, our survival, and of course our environment. Yet freshwater is also among the most threatened ecosystems, where wildlife has declined faster than in oceans or on land. And some challenges include climate change from global warming, where this affects freshwater habitats and all the local species and fauna that are unable to migrate or compensate for higher temperatures. And to make management of freshwater ecosystems even more challenging is that these threats often interact in ways that are often difficult to predict. And yet these threats should not be ignored, as it can lead to detrimental consequences and lead to poor decision making and lead to loss of species and biodiversity. So now you might be wondering how we can monitor these changes in river systems and so that we can react quickly to environmental threats in these regions. So this is where the study of the inhabitants of freshwater systems come into play. So you see, the study of our research focuses on a unique kind of organisms called macroinvertebrates. And typically this classification encompasses any aquatic animal and insect that are large enough to be perceived by the naked eye without any need of any microscopic equipment. They play a large role in freshwater ecosystems by recycling nutrients as well as providing food to higher trophic levels. Thus, these organisms serve as excellent indicators of the conditions of these freshwater ecosystems, as they spend all, if not most, of their lives in the water and are easy to collect and differ in the tolerance to environmental stresses. And this leads me to my research question. I want to explore how and to what extent microinvertebrates were being influenced by external stresses. And if we're able to understand the factors contributing to sustainable growth in macroinvertebrate populations, then we would be able to alter the environment or target our control and conservation efforts to help promote the growth of these macroinvertebrates to benefit these river ecosystems. And to quantify these changes, we decided to look into the effects of environmental gradients and the effect of predator populations of macroinvertebrates and their food webs. So we had two predictions. One, food web structure would increase in complexity with increasing distance from stream headwaters, where it's environmentally favored. And second, we also predict that top predator identity would negatively influence macroinvertebrate population body size, mainly as a result of trout-predator interactions, which we'll be monitoring by fish populations, as serving as a larger impact on average body size, more so than, say, the environment. But to answer my question, I first had to collect a nice sample of macroinvertebrates to study. So these samples were taken from the Sierra Nevada streams at 5 replicate stream networks, shown here on this diagram. The sites are also labeled very smallly, but it's in red along the streams on each of these diagrams here. And they're all taken using a DNet at these 30 centimeter squared sites, and then stored in 75% ethanol to be transferred back into the lab, where they're identified by the finest taxonomic level possible using a stereo microscope, and body sizes are also measured using these devices. And here we introduced a nice computer program called R, which helped us compute some nice computations. So spatial data taken from stream distances between sample sites were calculated using an R package called RiverDist, which utilizes data from the USGS dataset in order to determine pairwise distances from sampling sites along the river network. And with the collection of our raw data, we're able to evaluate some nice response variables. So we're taking a look at 3 response variables to test any changes and responses to our macrovertebrate samples. We explored functional feeding group densities, community weighted means, and food web metrics. And I will also be explaining what each of these response variables are in the following slides. Starting with the functional feeding groups. So as I briefly mentioned before, we know that macroinvertebrates, they serve a very integral piece in stream ecosystems. But as a community, they can be broken down into different groups depending on what role they play in the region. And the functional groups that are explored in this study are listed here. We have filterers, we have gatherers, shredders, predators, micropredators, and herbivores. And these functional groups generally occupy different microhabitats along the stream and can have different behavioral characteristics. For example, the shredders, which is studied here, they typically dominate, say, the headwaters and able to convert leaf decay into coarse organic matter, which is then filtered downstream and further processed by the filters. And data on the functional feeding groups were used from the EPA Freshwater Biological Trace database to identify sample taxa into their correlated groups. And more importantly, we're going to take the densities of all of these groups to then assess whether there's a correlation that exists between, say, the environment or based on the interactions with predator populations. And second, we have the community weighted means, which was our second response variable. It's also abbreviated as CWM. And the CWM evaluates the relationship between species abundance and their variability in body sizes among the taxa. And we use the site variability of the CWMs to evaluate how average community body sizes, so how large they are or how small they are, would be influenced by environmental gradients or by the interactions by the predators. And this metric was actually calculated using the dbFD function on the FD package on RStudio. And then lastly, we have food web metrics. These were identified using trophic interactions that was determined using known feeding interactions, which we identified using some literature that included observational studies, diet analyses, and stable isotope analyses. And we combined our data sets with these trophic interactions with those found in the function Well Builder on R to determine feeding relationships for co-occurring organisms found in each site. And from this, we're able to calculate three important food web metrics listed here. We used the trophic links, trophic link density, and connectance. And to further elaborate on what these three variables are, starting with the trophic links, also abbreviated as L. Trophic links is the number that represents all the possible interactions between the sample species. Linkage density, also abbreviated as LS, is another measurement used to understand the average level of specialization in a network. So, for example, if a network is dominated by multiple species with few interactions or multiple interactions, and this is typically calculated by taking the average number of links divided by the number of species. And finally, we have connectance, or C, is a measure of the proportion of interactions among all the possible ones in a network. And these are all three great indicators of the complexity of the food web, and also fluctuations in any of these metrics would indicate some level of response among the macroinvertebrates. All right, and now to get into the results. But before I go into the main results, I want to go over some of the statistical tests and measurements used to really generate some of these nice results in our investigation. Starting first with the PCA, or the Principal Components Analysis. This was a nice test used to measure environmental variables, which included some levels of dissolved oxygen, chlorophyll A concentration, pH, temperature, discharge, stream size, and land cover metrics. And these are all combined to quantify a nice environmental gradient, which we use to explore in each of the analysis, which you will see in the coming slides. And lastly, we also have these generalized linear models, also known as GLMs. And I provide a nice example of what a GLM is here in this nice table. But to explain, this is basically a nice method to really determine the best fit models. We'll be looking at a nice variable called the delta AIC value shown here, which is a nice indicator in which we can determine which model would best describe trends seen in our results. In the model shown here, we have four models looking at the environment on the effect of the sample sites. We also looked at fish, or the predator populations, the combined effects of the environment and fish, and also we have the null model, which is suggesting that there is no influence from either of these variables. And all of these models were determined using an R package, the AIC tab function, using the BBLME package on R. And this was just an example of a nice GLM, which is run for the community weighted means. And so with the delta AIC value, just to interpret what that means, with a delta AIC value of 0, that would suggest this is the most best fit model of all the four models it was tested against. And of course, these were run for each of the three response variables. And for the sake of efficiency, I just showed the final results of the GLMs, which I'll show you in the next slide. Starting with the FFGs, or the functional feeding groups. This was the diagram for our functional feeding groups, with all 6 of them listed at the top here: filterers, gatherers, herbivores, etc. And on the x-axis, we have, or the y-axis, we have the functional feeding group densities, and we represented them as a ratio, so as a nice little proportion against all the sites. And we compared them across whether there were sites present with the fish or absent, so whether there's predators or not present. And based on this GLM, there wasn't very decisive conclusions made based on these trends. For example, if you look at these four groups, you can see mostly the trends are pretty stagnant. So they weren't very significant, although we want to focus more on these two groups, the filters and gatherers, to really explain what's going on here. So we looked a little bit deeper and took a look at some of the averages, which was shown here on this little table. So these are the averages for all the density ratios, and focusing more on these two groups, separations based on predator population showed a significant result. For example, with the filters, you can see with sites where there are predators present, you can see there's a greater density of these filter groups compared to when there's no predators present. And the opposite was true for the gatherers. There's smaller density in the fish present sites, and there was a higher density for the fish absent sites. And this suggested that that the collector filters, or the filters in the gather groups, had shown a resistance to these changes based on the presence of the predators, while the other four groups, the herbivores, shredders, micropredators, and predators, showed little resistance or no change at all. Moving on to our CWM results. So these are the community weighted means. And this, from the GLMs, we determined that, that the model representing the environmental effects on the CWM was the best fit model. Here you can see on our x-axis, we have the PCA, which I mentioned before, the principal components analysis, which represents our environmental gradient. So values at the negative end on the left side represents more harsher environments, so example like lower temperatures, etc. And the opposite end, we have the positive values, more favorable environments. And then, of course, our community weighted mean is graphed here on the y-axis. And from this graph, we saw that there's a positive increase in the average community body size along the environmental gradient, which suggests that smaller body macroinvertebrates, so smaller values down here, were able to persist in more extreme value environments, shown by this best fit line. And finally, we have the food web metrics. The food web metrics were shown to be structured by both the interaction of the environmental gradient and fish presence based on our GLMs. And the most significant results we found from these two graphs on the left side here, showing linkage density and the number of trophic links. And we saw that in fishlet sites, shown here in the red, we saw for both trophic links and density, we saw there was a positive increase moving from environmentally harsh to favorable sites. And the opposite was true when the pattern was reversed in the fish absent sites. And that concluded our results for the three tests. And just to summarize our key findings for each of these tests, starting with the functional feeding groups, we found that predation negatively affected the densities for specific groups such as the collector filters, while stimulating the growth of the collector gatherers. And then for community weighted means, we saw there was a positive trend in the average community body size due to more favorable environments. And then lastly, our food web metrics, we found that predatory interactions combined with the environment were shown to result in fluctuations in trophic linkages and densities. And in conclusion, we found the results of our FFGs which supported the hypothesis that there's an influence of the top-down influence of predatory fish that overwhelms the influence of the environment. However, the environment does play an important role in monitoring and regulating the CWMs, and the environment and predator population jointly influence the food web complexity in our macroinvertebrate samples. And our findings suggest that there's a delicate balance in maintaining the integrity of food web structure and with consequences resulting in shifts for both environment and predator populations. And thus, the evaluation of the nature of the mountainous river ecosystems will offer extensive applications for maintaining species diversity, not only in river ecosystems, but many other organisms within those ecosystems, and conservation efforts for these land management in these regions. And I'd like to finally conclude with some acknowledgments for people that helped me with this project. Special thanks to my faculty mentor, Dr. Kurt Anderson, and big thank you to Matthew Green, who I believe is in the audience today, who assisted with a lot of the analysis and really introducing me to this project. And of course, thank you to all my family friends who are here present today. And I have my nice PBS statement. Thank you to viewers like you for coming out today. Thank you.